Okay, so Claire is the next speaker, and Claire has come to Telluride a few times. She was actually going to run a group together with Munya this year at Telluride if it had happened, and she's very interested in also in EEG and music. And so I'm looking forward to listening.
Hi, can you all hear me well?
Yes.
Okay. Oh, I cannot share my screen right now, weirdly.
Oh, one sec, let me...
Just need to make you a co-host, right? I can find you here. If you want, I can do it, Chichi.
No, I did before, but I think you left, Claire. Maybe that's why.
Yeah, I did.
Okay, yeah, you're a co-host now again. Can you do that now?
Yeah. Do you all see my slides?
Yes, and keep the questions coming. Somebody in the background will be answering your questions in the chat.
Okay, so thanks, hi everyone, and thanks for the organizer to invite me to give this talk, which I entitle Electrophysiological Markers of Timely Cognitive Processes, When to Use EEG. So this is going to be a rather general talk about everything you can do with EEG. So we're going to span different techniques, different studies, and I hope that's going to be giving you a good insight on how to use EEG signal. So before we jump in, as an introduction, we can talk about what is EEG. So EEG as a setup is a neuroimaging technique that collects electrical activity that is elicited by neurons from electrodes placed on the scalp. As you can see in this little figure, you have electrodes all around the scalp of the person, the participant. It is used both for clinical, such as epilepsy, anesthesia, as well as research purposes. Its main interest and quality lays into the very high temporal resolution that it has, which is an order of 10 milliseconds, or even better than that. And this offers a unique window in the evaluation and observation of dynamic cerebral functioning, or stuff that happens that are precisely timed. Okay. So then if we look at precisely what is EEG signal in particular. So EEG electrodes, they pick up on the summation of excitatory and inhibitory activity that are elicited by neurons. Here you can see the electrodes and the activations. The positive and negative poles are parallel to the neural orientation, as you can see in this figure here. And this polarity is elicited from very large groups of pyramidal neurons that are found in layer 3 of 5 cerebral cortex that are firing synchronously. Okay, so just before we jump in the actual core of the presentation, just a bit of history. So in 1924, Hans Berger was the first researcher to amplify the dipole activity that he collected from an electrode on the scalp. So this is the activity that you can see in this figure. So luckily, as opposed to what his lab looked like back in the days, EEG is very portable here, as you can see in this little figure. This is actually a portable EEG setup that you can just implement, and you see it's very, very light. Oops, sorry. So you basically just need a battery, an amplifier, a set of electrodes, and a head with a brain inside. And setting up an EEG lab from scratch would cost you approximately $10,000 or so. Okay, so I'm gonna span the main type of analysis that you can do with EEG signal. And for each one, I will first review the general method and signal processing behind it, and then I will give you examples, concrete examples from the literature of how that has been applied. So we're gonna go through ERP analysis, time frequency analysis, phase locking, and finally decoding and encoding, which is the specialty of the Telluride workshop. Okay, so beginning with ERP analysis. What is ERP analysis? So it consists in the fact that you present a stimulus, so it could be anything, in this particular example it's a syllable, repeatedly over and over and over again to a participant, and in the meantime you record the signal from a set of electrodes, so from an EEG. And what happens is that you take the neural response that is time locked to the presentation of the stimulus, and you do that for each iteration of your stimulus. And then you end up with like 100 neural responses that are time locked to your presentations, and you average this response. And by averaging this response, you remove some of the noise, and you end up with what are called ERP components, which are time-locked responses and peaks of amplitudes, either positive or negative, that are reflective of certain cognitive processes or certain neural dynamics, let's say. If you're lucky enough, you can actually try to localize these effects, or at least you can see which electrodes carry the most amplitude peak. And what is interesting with ERPs is that if you, for instance, if you present a set of syllables that are always the same and suddenly you present one that is different, as I showed in this example where you have ba, 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 da, then you will see in the response, in the neural response, change in amplitude in your neural peak that is actually going to be the marker of this cognitive processes that is, you know, encoding a regular stimulus and suddenly having to confront something different or something surprising or unexpected. So this component in this particular case is named the P300, and it's like very, very well known, very much used ERP that occurs at around 300 milliseconds. So moving on to an actual example from the literature, I picked this this science paper from the 60s, which is the actual first paper that mentioned the, or that, let's say, discovered the P300 ERP. So the text says the major waveform, okay, now I'm going to explain the setup, otherwise it doesn't make sense. So the study was as such, you had a cueing and then you had either a light stimulus or a sound stimulus. And there were two conditions. One that was like the certainty and the uncertainty trials. So in the certainty trials, the cue was showing, that was indicative of whether it was going to be light or sound for sure. And the uncertainty was cueing for the fact that it was going to be 50-50. And when you record the neural signal and then you separate, you average the trials and separate the certain condition and the uncertain condition, this is what you get. You get a neural response that differs very much between the two conditions. And you can see that the main difference is appearing as this negative peak here at 300 milliseconds. And the text says, the major waveform alternation is the amplitude of a positive going component, which is peak amplitude at about 300 milliseconds. So this is actually the first description of the P300 that has been all over the place ever since. Okay, so to conclude on ERP analysis, we can say that they're great for looking at neural responses that are time locked to the stimuli. Both the amplitude and the latency of the ERP itself is informative about underlying neural and cognitive processes. But ERP studies achieve noise removal, as I said, and pull out brain response by multiple repetitions of the same stimuli, as I showed with the da-da-da-bum, which is not very ecological and not adapted to continuous stimuli. Okay. So now we're going to move on to another technique that's called time frequency analysis. So before we move on to that, let me explain you that EEG data or EEG signal is composed of activity in separate frequency bands that are actually named with those Greek letters like beta, alpha, theta, delta, which are all well known and assigned to certain things. Like the alpha wave, for instance, is when people close their eyes and start to rest, they're going to show more alpha in their EEG. So what happened is that the EEG that you pick up in the end is the summation of all those frequency components in one big signal that has some temporal, that has some, how do you say, periodicity over different frequency bands. So what you can do, so yeah, EEG is composed of activity in different frequency band, and it is this activity in frequency band is non-stationary, so it fluctuates over time. And what you can do is using typically wavelet transform, you can decompose the signal into time frequency analysis that is going to describe the activity of this frequency into time. So in this particular study, they used such a time frequency analysis to look at the response to this particular stimulus paradigm. So it was like a semantically congruency priming effect. What they did is that they presented a sheep visual image and a sheep sound, like a bay, and same for a bell. That was either also an image of a bell and the ring going together. And there had two types of conditions. Either the image were matching, so there was like a semantic congruency between auditory and audio, or they were unmatching and it was incongruent. And the participants had to do some sort of like a task to evaluate either one or the other types of visual or auditory stimuli. And they looked at ERP first, and then they conducted this time frequency analysis, and they were able to look at the activity of the gamma band, which is the highest band that you look over into your EEG, which is everything above 30 or 30 to 70, let's say. And they looked at the time course of this activity and contrasted the two conditions, congruent and incongruent. And as you can see in this figure, they see a positive significant difference between the two conditions, showing that gamma band is actually has a larger activity time locked to around 100 milliseconds after the onset of the stimulus, in the case of the congruency matching condition. Okay. So what can we say about time frequency analysis? It's when you compare it to ERP, it's an enriched information because it's composed of both spectral and temporal information, where ERPs are actually just looking at temporal information. However, the type of transform that you do on the signal, which is typically wavelet transform or a discrete Fourier, how do you call it? Time Fourier discrete transform, whatever. Reduces the resolution on both dimensions, or you have some sort of a trade-off between the spectral and the temporal resolution. And besides, also another sort of problem, or at least caveat with time frequency analysis, is it usually falls into the framework of oscillatory activity and the theory that oscillatory activity are driving cognitive processes. And this framework, or this paradigm, is actually not very consensual in the community. So yeah, it's going to be more, it's going to take a further step than to interpret the data, because you're going to have to take a stand on this theory framework, let's say. Okay. Let's move on to a third type of analysis that you can do with EEG, and this is a phase locking value. So what you do when you look at phase locking value is that you estimate the phase value for each frequency channel and for each trial of your EEG signal. So you first apply Fourier so that you can decompose your signal, and then you apply a Hilbert so that you can compute the phase at each moment or each trial. And then you can average the phase values over trials and compare either two electrodes together, which is what this initial paper by this French team did, or you can actually also do exactly the same and compare the phase values between the one electrode and the stimulus, which is another way to do that that is also very popular. So in this particular study, which was carried out by the Flora Saneo in the Temporal lab, what they did is that they looked at the phase locking value between brain and stimulus during speech perception and production. So the paradigm was as follows: people had to listen to a stream of vowels, and then they were asked to simultaneously produce speech and with no specific instructions. So they had to say like a ta, 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 ta syllable while they were presented in their ears the syllables in rhythm. And what this team observed is that when you look at the phase locking value of the speech produced and the auditory input, you find a bimodal distribution of people, where you have on one hand people who highly synchronize to the auditory input, and on the other hand people who don't synchronize at all to the auditory input. And actually I have an example to show you of the two types of participants. So this is a typical low synchronizer. I don't even see anyone. Can anyone tell me if they hear well?
Yes, I hear you. I mean, I heard it.
Okay. And
Yeah.
This is one high synchronizer. So another participant that is typical of the second peak in the bimodal distribution. You can see that in the second case, the speech is much more regular and feels like it's tracking something rhythmic. So then they took some participants from the two groups and they put them in the MEG. So it's not EEG, but it's almost the same, let's say. More expensive, more fancy, but you can, in terms of phase locking value analysis, you can do the same thing. But they put them in the EEG and they looked at the neural signal and how well it was phase locked to the auditory input. So this time they didn't have to do anything. They were just listening to the stream of syllables, of rhythmic syllables. And the team performed a phase locking value analysis on the EEG signal and looked at how it was phase locked to the stimulus. And what they found is they found a similar dichotomic distribution or bimodal distribution between high and low synchronizer, not in speech production, but actually in the way their brain was entrained to the auditory stimulus. So here in the red and yellow, you can see the difference between the two groups and where it was located in the brain. Okay. So the phase locking value reflects the entrainment of a neural response to a particular stimulus. It tends to work better when there is a clear peak in the frequency spectrum, such as the one you have when you present a regular stimulus. And if not, you need to extract the frequency band of interest and then perform the phase analysis. So in this case, well, as always, but especially, you need to be very mindful about the baseline condition and carefully shuffle trials so that you have a good permutation test to test your actual phase locking values against. Okay. So now I'm going to move on to decoding and encoding, which is the fourth and last type of techniques, EEG techniques that I'm going to present. So the idea of decoding and encoding is quite simple. It's the idea that when you present a stimulus to a participant, to a brain, the stimulus has some characteristic or some features that you can represent or extract. And here, this is the waveform, the sound waveform. And the idea of encoding is that your neural response is going to contain some sort of imprint of this specific stimulus feature somewhere hidden in the stimulus. Somewhere hidden in the neural response that you're going to pick up with your EEG signal. And the decoding or encoding approach is to sort of see how well, the decoding approach is to see how well this particular feature is represented into your brainwave, whereas the encoding is going to be to reconstruct the stimulus from your brainwave, which is a bit different, but anyways. So in this particular study that we conducted with people from, First, I'm going to present you one particular technique of decoding, which is called TRF. So TRF stands for Temporal Response Function, and it's an approach that we took when we conducted this study on looking at the encoding of musical features in the brain of people. So the one thing that I didn't mention earlier is that when you do decoding, you can either look at very stimulus-driven low-level features such as the envelope, but you can decode potentially everything or anything that is structurally meaningful, however abstract. So in this case, we were wondering about how well you can decode musical syntax or musical expectations from the brain of people. So what we did in this case is that we applied TRF. In this way, we first had people presented with some musical stimuli, and I'm going to play you an example right now. There were Bach monophonic arrangements of okay. So from this type of stimuli, we derived two features. First, we derived the envelope, easy, and then we derived, easy, still, and then we derived what we call the surprise value, which was a reflection of the musical expectancies or the expectations or the musical syntax, if you want. And the way we extracted this the surprise value was by using a model called Idiom, which is a model based on variable order Markov chains that looks at the typical sequences of notes in a large corpus and in the specific musical stimulus that you have, and then predicts the surprise contained on each event. And this is very similar to stuff that you have in language, where, for instance, if you have I'm going to go to the dentist to check my, and then you have a value that's going to be larger if your next word is hair, and a lower value if your next word is tooth, because it's more or less expected. Same thing with the music, same thing with the surprisal model of this music. Okay. So what we do then is that we apply a ridge regression linear model to see how well the features, those two features, the envelope and the surprise, were encoded in the EEG signal. And to do that, we apply the TRF algorithm, which fits a filter or a kernel that is going to best describe the mapping between the set of inputs or the set of features and the neural response. And then you use this filter to predict an unseen part of the EEG. And this unseen part of the EEG, then you correlate with the actual EEG. And this correlation between the predicted from your filter and the actual is going to be your cortical tracking index, if you want to call it this way, or your mapping quality representation of the feature, something like that. And what we did in this study is that we contrasted two conditions. One was when we took only the acoustical inputs, so only the envelope. And the other one was when we took the envelope and the musical surprise. So it was like acoustic plus musical surprise. And the hypothesis is that if the surprise is indeed we can decode it from the EEG signal, then we would have an enhanced cortical tracking from this model, because it contains an information that is actually imprinted in the EEG signal. Okay, so here are the results. We had EEG for that, 20 participants, 10 musicians, 10 non-musicians. So first we didn't look at the group effect, but we just took everybody together. And this is the main result summarized in its simple form. It's that the prediction correlation, which is equivalent to the cortical tracking, is actually higher when you look at the model that contains the musical surprise as opposed to only the acoustics. And this is tested against permutation within each condition. So then you have the same figure, but broken into different participants. So in red, you have only acoustical model. In blue, you have acoustical plus surprise. And those gray bars here are the difference. And you can see that basically the difference is higher than 0 for almost everyone, which means that at the individual level, we can also track the musical surprise from the EEG signal of individual people. This is just a topographical mapping of where the main effect is. Nothing much informative about that. So it's picked up by central electrodes, which are typical of auditory responses. This is interesting. This is a control sort of analysis that consisted in taking the idiom model that we use to generate the surprise and reduce the window, the memory window that it was using to make the prediction. So reducing the memory window would mean that you take less and less information to make the prediction about your next node. So the smaller the window, the less informed or the less precise is going to be your prediction. And when we take this actual altered model and we put it as an input for our TRF, what we observe is that we have actually a decrease of the correlation or the cortical tracking, meaning that the actual input matters into how well we can decode the information, with the most informed input giving the best results possible.
Claire, can I ask a question on your middle plot? It says non-musicians, musicians, but I don't see anything on your plot that indicates.
Oh, sorry. Yeah. So those are the full and the non-full circles here. Do you see that?
Yeah,
it's very subtle. Actually, okay. I'm moving on to that. It's the next slide.
Okay, perfect. Thanks.
So yeah, here it's not very well enlightened, the difference between musicians and non-musicians. Here we actually broke it into two groups. So this is a group analysis where you have the results for non-musicians and musicians. And the take-home message from that is basically that melodic expectations is larger for musicians than non-musicians. And this difference is mainly picked up by frontal EEG channels. So, which is totally in line with previous literature that has shown that musicians and non-musicians are almost the same, just that musicians are usually typically a little bit better or a little bit more precise about their predictions. Studies about reaction times showed that already, or even ERPs or things like that.
Is that Shihab Sharma on the paper?
Yeah, that's him.
It is. So
hey, Claire.
Yeah.
Claire, two questions. First, someone asked if surprise signal encodes the peak in the signal.
Sorry, what?
Someone asked if surprise signal encodes the peak in the signal.
The peak? What do you mean?
Yeah.
The peak? The onset.
Yeah, I think call it the onset. Yeah.
Yeah. It's time locked to the onset of each notes.
Okay, so it's reasonable to say that the surprise signal is encoded by the brain there. Or you're picking up the surprise signal in the responses. Is that fair to say?
I'm not sure what you're going to say. So the envelope and the musical signal are very much alike, because basically the envelope shows peaks at each note onset, and the surprise is one single value at each note onset that is larger or smaller, just depending on the surprise value of it. So they're highly redundant, which is why you have to carefully apply permutation tests to validate your findings.
Cool. And then going back a couple sections in your talk, someone is asking if there's a reference that talks about how spike timing at the neural level correlates and becomes an aggregate time frequency measure. Do you know of any literature that talks about that?
Going back to the time frequency?
How you go from spikes to a frequency response, a gamma response.
Oh, wow.
Sorry, big question.
I just know that in theory, oscillatory activity are supposed to not even reflect spikes volume. They're supposed to reflect higher or lower level of excitabilities of neurons. So you're very, very far from the spike or the rate base or whatever signal. Is that answering any question?
I think that's as good as we can do right now.
Yeah.
I'll let you continue.
Okay. Okay, so just concluding on the decoding and coding stuff. So it's great for neural response to variable, continuous, multi-dimensional stimuli. So in one word, eco-valid stimuli, stuff that we actually listen to in the real world. The downside, in one way, is that you depend heavily on the input, which corresponds to the feature extraction model. So as I said, when you are extracting envelope, then you are you know pretty well what you're extracting and how to extract it the best and everything. But when you're, for instance, decoding melodic expectations, of course, your model, your results rely heavily on the quality of the model that you're using. So is this the best predictor of what's actually going on in the brain or not? So it's like adding another parameter of the problem here. So, and finally, for, with decoding and encoding, you want to be always very mindful of overfitting, and to keep in mind that you want to think about a very good baseline that you're going to contrast your main result with. Yeah, I just want to point those different papers that are also looking at decoding, if they're enveloped for the two first, or highly abstract information such as the semantic content of sentences for the last one, which are all Telluride babies in one way, because those people are Telluride people. So yeah, you're invited to check those out. Very interesting papers. So to conclude on EEG signal and what you can do with this, so one point to keep in mind is that it's in all hands that you make the best broth. I don't know if it's an English saying, but it's a French one at least. And EEG is almost 1 century old, and it's been very useful for the past century. So it's one good side that it has on this side. It's also portable, low-priced neuroimaging technique that has a very precise temporal resolution, as we said, on the order of 10 milliseconds. There are a variety of approaches depending on both the stimulus, the design, and the question or the framework that you're putting yourself in. And of course, it's because of its temporal resolution, it's very efficient for looking at the response to dynamical stimuli, which are, for instance, sound, speech, and music are very well fitted for EEG analysis. Okay, I'm done.
All right, thank you. Thank you, Claire. It's very nice. Yeah. So Malcolm, did you have some questions from Slack that you?
I think we've got everything covered. I guess the one question that's still pending and maybe is worth talking about is how do you establish how much of the effect you're seeing is because of surprise versus just the intensity signal? That seems to be key to the paper you were talking about, Claire.
You don't really quantify it, I guess. You just look at whether there's a significant difference between the two, and that will just, well, actually, yes, you do, because then you can look at, when we look at musicians and non-musicians, we see that the increase of musical surprise is actually higher when we compare to non-musicians. I'm not sure what the question is. I mean,
I think that's fair.
Okay.
I mean, I think we're seeing a correlation with experience. So if a person has a musical experience, then their surprise signal is more useful.
We didn't actually look at correlations because in this paper, we took 10 professional musicians and 10 totally naive musically people. So we don't really have a range to look over, but that would be certainly something.
Up to.
Yeah.
Cool.
Is it possible to train somebody to the, say, correct form of the music? And then, because as I remembered, you took a piece of music and then you did something to it.
That was, that's probably going to be upcoming Telluride's topics.
Well, we did that last year in Telluride, didn't we, with teaching people Chinese or Indian music?
Yeah, exactly. Yeah. Now we're collecting data on that on the MEG here, and what we do is that we have people, we have a set of stimuli that are Chinese melodies. And we have people, so those Chinese melodies are from a corpus that has been studied by the guy who actually designed EEG, which is the model for extracting surprise. And he showed that the surprise that you extract from this corpus is different from the one that you would extract on the same melodies if you were training your model on Western music. So meaning that the grammar of this corpus is different from the grammar of Western music. And what we do is that we have people come in the lab, they listen to a set of those stimuli, of those Chinese melodies, then they will go home and they listen to 10 hours of the larger corpus that we have. And then they come back again and they do the same, they listen to the same exact melodies and another set of unknown melodies, but still from the same corpus. And we just track the enhancement of how well they can, we can see the surprise, the grammar from this corpus in their brain before and after. So we sort of track the learning of the grammar. I don't hear you, Chichi.
So the EEG signal usually has a lot of artifacts, right? Which you didn't really mention. So
that's true. Yeah,
quickly on the kind of artifacts you would see.
If you have good participants, right? You don't have that much artifacts. So yeah. So that's one thing, is that you want to harass your participants into staying completely still throughout the experiment. And if they move, you have to cut out the. You can also apply different techniques to remove regular artifacts, or artifacts that are going to affect different channels, such as eye blinks or things like that. Yeah, that's true. That's the pre-processing part of it.
Okay, that's great. Thank you. Any other questions, Malcolm? Anything?
Lots of little things in the chat. Nothing I want to bring up if we're running late on time.
Okay, all right.
I have a
comment. This is Andreas. Just a second. Claire, thanks. This is really a wonderful presentation. Thank you for joining us.
Thank you.
And oh, okay. So Terry, you had a
quick question. So you showed us, the peaks by themselves and the peaks plus the surprise. What about surprise by itself?
Yeah, that's a good question. Surprise. I'm not sure. In principle, you can totally do it. Actually, there is one study going on with Imagine Music, who uses exactly this technique, where you don't even include the envelope, and you should have a pretty faithful tracking already, because almost everything contained in the envelope is contained in the surprise signal as well, which is the onset information.
Okay. All right. So thank you.
One last point about that.
Oh,
you can do that if you can contrast two sets of surprise, for instance, like what we do with the enculturation, where we have before and after. But if you don't have those sort of a contrast, which is the case here, you just look at whether you can decode the surprise, and you need a baseline that's going to be just your envelope or your shuffled surprise, for instance, or something like that.
Good, Terry.
Good, thank you.
All right, thank you, Claire. Okay, so I'm going to stop the recording.
Thank you.
Thanks. Okay.